Hi guys, and welcome back. Talking a little bit today about macromolecules. So macro meaning large. So large molecules. So you probably, or you might be familiar. I said probably before, and someone said I haven't ever learned about this. So macromolecules. So they're large functional carbon-based structures. Carbon, right, is the building block. So we're a carbon-based life form. So all of our important molecules have carbon as our foundational building block. So there's 4 basic types, and you might recognize 3 of them from food. So we have carbs, lipids, otherwise known as fats. We have proteins, and we have nucleic acids, which you may know as DNA or RNA. Okay, so we'll get into it. All right, got a lot to get through, and you probably don't want to sit for an hour. All right, so macromolecules, they're made of monomers. All right, so mono means one, right? So mer is kind of like body, right? So each monomer can be joined to make a dimer. If I have 3, I'd have a trimer because di means 2 and tri means 3. And many makes a polymer. So the macromolecules are all polymers because they contain many pieces, right? So let's just get right into it. So carbohydrates we'll do first. Right? So we'll do these carbohydrates first. So carbs are sugars, right? Important things to remember. They're the main source of energy, right? So for us, glucose so we get energy from glucose, right? No matter if you're eating fruits or if you're a baby and you're nursing, we got to convert it all to glucose before we make energy out of it. Right. Okay, so sugars can be monosaccharide. So this word, let me just expand this a little bit. All right. So a monosaccharide, it's a monomer. Is a monomer of carb. So saccharide means sugar, right? So a saccharide is a simple piece of sugar. Simple sugars are monosaccharides and disaccharides. So di would be 2 put together. So all monosaccharides have this formula. Kind of important. I probably won't test you heavily on that. CH2O. So it's a ratio of 1 carbon to 2 hydrogens to 1 oxygen. So for instance, glucose here is C6H12O6. So I have a 1 sugar to 2 hydrogens to 1 oxygen. So same ratio as that right there, right? So common forms. So you need to know these names: glucose, galactose, and fructose, right? Remember, glucose is where we get most of our energy from. Galactose would be from in milk, right? And fructose is from fruit, right? So all have the same formula, right? Are all called isomers, right? So isomer, right? Iso means same, right? So they all have the same formula, but they're arranged differently. So if you look at glucose here, galactose here, and fructose here, they all have the same amount of carbon. They all have the same amount of oxygen and hydrogen, but they're shaped a little differently. The glucose and the galactose look really similar, right? But the difference is where this group is placed, right? So just a small little change, and we can make those changes in your liver. So if you had galactose, it gets sent to your liver, and the liver would just flip that bond around, and the galactose would become glucose. Same thing with the fructose. But fructose, you got a little more changes that have to happen, right? We have to disattach this oxygen and reattach it over there, right? And then we got to switch a couple things around, not a whole bunch. We got to flip these structures right here, and then we can make fructose look like glucose, right? But it all has to be changed to glucose before I can make energy out of it, right? So make sure you know that glucose is the main source of energy for making what's called ATP, which we're going to use as energy we'll talk about later. So synthesis means to make. So the making of carbohydrates, right? So each can be formed, important things, each can be formed through a condensation reaction, which you may know as dehydration. Synthesis. Right. So dehydration means to remove a water, and synthesis to make. So I make by removing the water. Let me write that over here a little better. Let's try to squeeze it in there. So we'll write that over here again. And condensation means to remove water as well. So commonly called dehydration synthesis. So if I take two monomers of glucose, so I have a glucose there and a glucose there, you'll notice that they both have these OHs. So OH is a hydroxyl. You need to know that because you're going to hear that in a lab video. So it's a hydroxyl, right? So they each have these hydroxyls. So if I have a hydroxyl here, and I have a hydroxyl here, right, I can take this out, which is H2O, which is water. So if I take that out and I join them together, right, I've removed the water, which is dehydration synthesis. Right, so I have these two hydroxyls, right? I remove that water here, and now I have this bond. And this bond is going to be called glycosidic linkage, or simply a glycosidic bond. Right, so the removal of water. So if I can form it by removing water, I can separate it by adding water. So hydrolysis, addition of water to break the bond. So if I can take this disaccharide here, sucrose, and I add a water in here, I can separate it. So sucrose, which we'll talk about, is common table sugar. So common table sugar has a glucose and a fructose together, right? So you can take this table sugar apart, you can use the glucose right away, and that fructose you can send to the liver to make glucose, and then you can use that as energy. So you need to know these names. So you should be writing these down. Common disaccharides, maltose is glucose plus glucose, sucrose, glucose plus fructose, and lactose, glucose plus galactose, right? So those are common disaccharides. You need to know those. Common polysaccharides. So remember, poly is many put together. Right? We have starch. Right? So like a potato, right, is just a big ball of starch, and that's plant storage, and that's many glucoses stuck together in a chain. So when you eat a potato, you can just start taking all those little glucoses off it, and you can use them as energy. Right? Cellulose. Cellulose is a lot like, so this is branched, this is linear, which you'll see when you do your lab video. Cellulose, because it's linear, right, and they kind of make all these molecules stack up really well together. So since they stack up really well together, I can't get enzymes in to take them apart. We'll talk about enzymes on Thursday. So they're not digestible to humans, right? And then glycogen. So glycogen is animal storage for food. So where a plant will store starch, you'll take your glucose, your excess glucose, and you'll store it as glycogen. That's another branched structure, right? But that's the storage for animals. So make sure you know what these three are. There's others. There's chitin, but those three are the most important ones we need to know for now. All right, so we'll switch from carbohydrates to lipids. So lipids, otherwise known as fats. So those would be your oils, right? Your waxes. They're connected by ester linkage. So remember that carbs use glycosidic linkage, and then fats use ester. So an ester has a carbon connected to an oxygen connected to a carbon, right? So that's a ester right here. Right, ester means fat. Okay, so know your parts, right? So this is a glycerol, and then these are fatty acids. Right? So these are made of carbon, hydrogen, and oxygen, just like carbohydrates, but the ratio is different. Okay, so we're not tied to that 2 hydrogens to 1 carbon and 1 oxygen. All right, so we have 3 glycerol here, and we have a bunch of fatty acids. All right, and you'll notice that it's the same mechanism. Right? So these are all formed by dehydration synthesis. Right, so they're all formed. So they're all formed, right? Here's my 3 water removals right there. So they're all formed by dehydration synthesis, and I can all take them apart by hydrolysis, just like the other one. All right, so we have our glycerol we talked about, right? We have our fatty acids, right? These are very hydrophobic, right? Oil and water, right? Doesn't mix, right? So they're hydrophobic, or they're repelled by water. So we always say water hater, but it's repelled. So that means repelled by water. Okay, so those fats, lard, butter, saturated is solid at room temperature. They could be tightly packed, or so. But here's the thing. So here's my fatty acid here. If it has no double bonds, it's saturated. I'll use a different color for this because so if it has no double bonds, it's called saturated. So saturated means holding as many, right? So saturated means it's holding as many hydrogens as it could possibly hold, right? And over here, it's unsaturated. So you may have heard of monounsaturated. So this unsaturated means it has at least 1 double bond, right? So this would be a monounsaturated, right? Polyunsaturated would mean containing many double bonds, right? So let's make sure you know that. A fat with at least 1 double bond is said to be unsaturated because it could still hold another hydrogen. Right, so things that we use lipids for, storage of energy. So if you eat excess calories, you store energy in the form of lipids. And I can take a carbohydrate apart, right, since it all has carbon, hydrogen, and oxygen, then I can reformulate it into a lipid. So if you eat extra carbs, you'll store them as energy, right? So we store them as fat or adipose. Adipose means fat tissue, right? We make cell membranes out of it, right? We'll talk about cell membranes. So you need fat in your diet. If you don't have fat in your diet, your body will make it. Right? We use it as chemical messengers or steroids, right? Very important to your body. You use it as insulation and shock absorption. Most of your brain up here is adipose tissue, right? Keeps your brain warm so it functions well. Shock absorbs it for when you knock yourself in the head. It surrounds your heart, right? You've got a fat layer surrounding your heart. So most of your organs, you have a fat layer surrounding insulation and shock absorption. Right, proteins and nucleic acids. So proteins make about 50% of the dry mass. So if I took water out, remember you're about 2/3 water. If I took the water out, right, 50% of everything else would be proteins, right? Proteins make up enzymes. So for biotechnology, enzymes are super important. We use enzymes to do all our work. So enzymes speed up chemical reactions, right? So enzymes speed up chemical reactions. So nothing would happen in your body without enzymes. So when we talk about dehydration synthesis, making molecules, right? Dehydration synthesis is not possible without enzymes. Hydrolysis takes them apart. Hydrolysis is not possible without enzymes. So I'll talk about enzymes the whole day. Use proteins for storage, defense against foreign substances, structural components, transports. Probably a big one for you is muscles are made mostly of protein, right? And then we do some cellular communications with it. So we use proteins for a bunch of stuff, right? The really important parts here, enzymes, right? Those are the important things that we're going to work about. Okay. Peptide bonds. So a peptide bond joins amino acids. And amino acids are the monomers of proteins. Right? So they made peptide bonds join amino acids. Amino acids. So proteins are made of amino acids, which are joined by peptide bonds. Right? So all amino acids have the same formula. So we have N, we have C, we have C. All right, so R, this is my functional group. This over here, bring it down. This is an amino. And this is what they call that's what they call it, an amino acid, because it has an amino group, right? It has a carboxylic acid group, and this is called the alpha carbon. That's a symbol for alpha, and that's where the functional group is. So all the proteins have the same backbone, but the R makes them all different, right? 20 different amino acids, right, make up all the proteins. So I have 20 different functional groups, right? So we say that amino acids are joined from N terminus, that's your amino group, to C terminus, which is your carboxylic acid, right? All right, so same thing, right? So to put them together, right, I got H2O. So again, this is joined. Joined via dehydration synthesis, and if I wanted to take them apart, I would do hydrolysis again. All right, so there are 20 amino acids that make up all of the structures in your body. So do not memorize this, so I can have non-polar or I can have polar, right? So non-polar, these would be hydrophobic, and polar would be hydrophilic or attracted to water. Hydrophobic means repel, right? Okay, so that's probably what you need to know there. Protein structure, right? So you need to know your structures. A lot of notes to take. So primary is just a sequence of amino acids, right? So when I put them together by dehydration synthesis, I make primary. So structure, so once we start making proteins, they'll start to fold, right? So I'll either, this should say alpha, but Google doesn't like that, right? So I'll either make a beta pleat, right? There we go, I lost my pen there for a second. So I'll either make a beta pleat, which is just a folding, or I'll make an alpha helix. So alpha helix are circles, right? So that's my secondary structure. So my primary structure will not work as a protein. My secondary structure will not work as a protein. My tertiary structure. So, right? So now I got a 3D shape. Right? So this 3D shape is also known as it's a conformation, right? And that is the functional what's called a functional protein. Right, so all proteins at least, right, make sure you know this. So all proteins at least have to have a tertiary structure, which is conformation. And that conformation is going to be really important because that conformation is going to be what makes enzymes work, and without enzymes you can't live. Quaternary is just 2 or more, so these would be what they call globular proteins. So some proteins are globular proteins, many are, but globular proteins are 2 or more tertiary proteins put together. Okay, but all proteins need to have at least a tertiary structure. You need to remember that. Right. So conformation, by the way, this is affected by temperature and pH. Remember, pH is the amount of acidity. So our body, when we say, you may have heard the word homeostasis, and homeostasis is a process where we maintain our internal environment, so we maintain protein structure. So if you get too cold, your proteins start to wind too tightly and they don't work. You get too hot, they start to unravel and they won't work. That's why if you get a high temperature, right, once you get about 101, 102, 103, we got to get that down, because once you hit about 105, your proteins get loose, you start to have seizures because your proteins aren't working well anymore. Same with pH. If your body gets too acidic or too basic, right, your proteins will unravel or be bound too tightly and they won't work either. Right, so it's just an introduction. Nucleic acids, we'll have a whole chapter on DNA, so I'll just give you the basics of it, right? Nucleic acid, 2 basic types, DNA and RNA, right? So DNA, remember, is the genetic code, right? Each of our cells has all the DNA needed for our body, right? And RNA does the work of DNA. Okay, so 3 parts, right? For nucleic acid, there's a 5-carbon sugar, a phosphate group, and a nitrogenous base, right? So if I looked at it, it would be, right, and then here's my R group. And then, so this is my 5-carbon sugar. This is my phosphate group. And this right here is my functional group. A, C, T, G. The 4 functional groups that make up DNA, right? You could have uracil instead of thymine if you were RNA instead of DNA. So that's basically it, right? And our bond is going to be a phosphodiester bond, but I'll talk about that later. Right? So that's it for today, and I'll talk to you soon.